Okay, cool. So let's go ahead and get started. So thanks everyone for joining this class. It's the first time I've taught it, and I'll be kind of developing some of the course materials along the way. So yeah, it should be exciting. So yeah, basically this class is a freshman seminar that I'll be teaching you about some of the modern techniques for how people track viral epidemics in the day of modern sequencing. So I'm gonna share my slides. Let me get my share screen going. Okay, so hopefully people can see my slides. I'm also gonna pull up the chat just in case people have questions. So at any point, if you have any questions, feel free to type them in the chat. And you can select all panelists and attendees from the dropdown just to make sure that everyone can see it. But yeah, cool. So first, I wanna just kind of introduce myself and introduce the class. So for people that might not know me, I see some familiar names, but just for everyone else, my name is Nima Moshiri. I'm an assistant teaching professor in the computer science and engineering department. You can call me Professor Moshiri if you want. You can just call me Nima. Anything's fine. So my research area is in bioinformatics. And specifically, I focus on developing computational methods and statistical models for viral epidemiology. So historically, I've mainly worked full-fledged in COVID-19. So a lot of the return to learn stuff that you're seeing, I'm very integral in a lot of the data analysis side of things for that. So fun stuff. So as far as teaching goes, I have taught intro CS, I've taught data structures, I've taught algorithms, I've taught some bioinformatics classes. Currently, I mainly teach CSE 100 and then also this seminar class now. And then here's just a full list of classes I've either taught or TA'd for. So I'm also very active in developing massive adaptive interactive texts, or MAITs, as well as massive open online courses. So I'm very interested in online education. If you're curious about that stuff, feel free to hit me up. And just kind of lastly, my hobbies, I like video games, movies, TV shows. Right now I'm playing Final Fantasy 15, which is a lot of fun. I like the Fast and Furious movies. I like Lord of the Rings. A little bit about me. So before I go into the course topics for today, I'm gonna just quickly pull up the Canvas page, just so that everyone's on the same page. So if you go to Canvas, it should look something like this, hopefully. Oh yeah, thanks. Yeah, people noticed the Hobbit hole. So the homepage of Canvas should look like this. We're gonna have 8 weeks of instruction, so you won't have any class weeks 9 and 10. And basically, the class is pretty much just attendance based. Like you don't even have to technically attend. I'll be posting recordings of these sessions here. But instead of attendance, you're going to have these weekly reflections that, they're not really graded for any correctness. It's just, I want you to reflect on what you've learned in the past week's content and maybe summarize it, talk about what interested you, just somehow interact with what I talked about that week. So each week you should submit your weekly reflection based on that lecture by Sunday. If you submit it late, that's also totally fine. I'll be flexible just because this is a seminar class. But yeah, so every lecture, I'll be posting the lecture slides under that given lectures tab. And then after that lecture, I'm going to post a recording of it directly under the slides. So I haven't posted any of the other lecture slides yet because I haven't done them yet. But so Anisha asked, a little confused, should the reflection be submitted if we don't attend class, or should we always submit? So if you don't attend synchronously, you're expected to watch the recording afterwards. So everyone should be attending in some fashion, either synchronously right now, or you can follow up with the recording afterwards. Or you can just click through the slides if you prefer. But yeah, everyone should be submitting one of these every week. Cool. So the syllabus, I just kind of go over some of the learning objectives. We're going to talk about some public health questions, how we use molecular epidemiology to explore these questions. And I'm going to go over kind of like a brief overview of some of the state-of-the-art tools used for each of these different topics. It's a 1-unit course. There's not much work. It's mainly just for all of you to learn a little bit. So basically 2 hours per week, 1 hour will be either synchronously attending this presentation or watching the recording. And then the second hour is just, writing that reflection, just thinking about what you learned during that lecture, stuff like that. So this class is pass, no pass, and to get a pass, just finish all the reflections. I can make exceptions for special circumstances, but just try to get to all of them. So someone asked in the chat, how long should our reflections be? like a paragraph. If you want to go longer, that's totally fine. But even just a handful of sentences saying like, I learned about this cool topic and this was what I found interesting about it, would also be totally fine. Just something to help you engage with the materials a little bit. And then the assignments, it's just the reflections, which you can find those links here instead as well. And then grades are just purely the reflections. And then there's the discussion board where if you want to chat with your peers, feel free to post on the discussion board. I don't check this super regularly. So if you have questions for me specifically and you ask a question on here and I don't respond to it, feel free to email me. And I can, just ping me and I'll check the discussion board. But cool, any other questions about course logistics before I jump into the topic for today? It's like a pretty chill course. Basically show up and then submit the reflections, and that's all of it. My main interest is just having all of you learn a little bit. Cool, okay, it seems like there's no questions about logistics. If you have any questions later on, feel free to type them in the chat. But cool, let's jump back to the materials. So today's goal, as far as course content goes, is that I want to just kind of introduce everybody to the world of epidemiology and public health. So this is a super, super quick intro. It's definitely by no means what you'll get from an actual epidemiology course, but just kind of like the TLDR that's pertinent to what I do. So first, public health. So public health is the science of protecting and improving the health of people and their communities. So in general, a standard public health approach for responding to a problem, we have some problem that exists and we want to reach some response that will be the public health intervention that solves that problem. Generally, the first step is surveillance. You want to identify what is the problem. You want to describe it concretely. You want to really figure out what exactly is the public health problem that we have. Then second, after you've done surveillance, the second task is risk factor identification. So what is the cause of the problem? What is causing people to have this issue, or what is making it a problem in my society? Then after that, the third thing that you want to ask is, you want to do the intervention evaluation. So what works? If I'm trying to tackle those causes, what specific interventions help, tackle those causes? And then lastly, implementation. How do you actually do it? So once we've determined what the problem is, discussed what is exactly causing the problem, thought about what types of things we can do to try to, intervene and fix those causes, the last thing is how do I actually implement those in practice? And implementation, you have things that have to do with like how many resources do we have, what populations do we target, all that kind of stuff that actually realizes it into reality. And I'll be monitoring the chat throughout the talk. So if you have any questions, please feel free to just type in the chat. Cool, okay, so that's public health. So another concept that I want to introduce is the world of epidemiology. So epidemiology is the study of the distribution and determinants of health-related states among specific populations and the application of that study to the control of health problems. So basically, the study of what health-related things are going on in my population, and also applying that knowledge to the control of those problems. So it's kind of like a two-pronged study and apply approach. So one question that's super important for us to ask is how do we actually utilize epidemiology within public health practice? So for public health practice, we define that 4-step plan. How do I actually use the study and application of disease-related states to those 4 steps of like solving real-world problems? So the first thing that epidemiologists try to do is discover the agent, host, and environmental factors that affect health. So this is very similar to that first bullet from that previous slide. You want to discover just what are the different participants, what are the different factors that are coming into play here. And then you want to determine the relative importance of the causes of illness, disability, and death. So for example, a given illness might have multiple different causes. Some of them can be biological, some of them can be socioeconomic. And you want to determine out of all of these causes of some real world issue, what is the relative importance of each of these causes? Right. So for example, like having chronic back pain, there's probably some biological and like genetic factors that go there. But then there's also things like, do you have money to afford a nice bed that has proper back support? Do you have a nice chair or work environment? Stuff like that. So there's biological and sometimes socioeconomic and other types of, social science factors that also contribute to illness, disability, and death. All of those are equally important, and you have to figure out what is the relative importance of each to this issue. And then identify the segments of the population that have the greatest risk from those specific causes of ill health. So once you've identified, maybe you can't afford a high quality bed, what segments of the population are most at risk of that specific cause? Stuff like that. And then evaluate the effectiveness of health programs and services in improving population health. So basically, once you've designed programs and services that target those specific issues in those specific subpopulations, actually evaluate how well are those things working. Maybe measure what is the number of reported chronic back pain issues that you see in your population? What is the incidence of some disease, number of deaths due to some disease, stuff like that. Actually evaluate how effective was your intervention at preventing this. Cool, any questions so far? Kind of makes sense. Cool. Okay, so that's kind of, there's a hand raised. The hand went down. Did you want to ask a question? I can always unmute you if you want to ask questions verbally. I don't know if that was just an accident. No? Okay, cool. Let's see. Let me get these things side by side. Okay, so the next area that I want to talk about is now a little bit more specific to my own field. So now the next thing is more specific to my field, which is phylogenetics in an epidemic. So as I mentioned, I'm specifically interested in viruses. Let's talk about how evolution and epidemics play hand in hand. So first, just because I assume that people maybe haven't taken a bio class, let's go over a review of phylogenetics. So a phylogeny is just a very fancy word for saying evolutionary tree. It is literally synonymous with the term evolutionary tree. So if you hear phylogenetics, that's just the study of evolutionary trees. So here we have an evolutionary tree composed of human, chimpanzee, gorilla, and orangutan. And in general, with an evolutionary tree, the present day species represent the leaves, those bottom tips of the tree. And then the internal nodes, the internal kind of points in the tree are the ancestors. And in general, the present day stuff are currently alive, and ancestors we generally assume to be extinct. And then not only do we have the shape of the tree, but we also have lengths on each of these branches, where the length of a branch is some unit of evolutionary time. That could be days, it could be weeks, it could be years. For this order of magnitude of like orangutan, gorilla, chimp, human, it's probably like millions of years. But for viruses and stuff, it might be days, you might measure it in number of mutations, different metrics that longer distance corresponds to more time has passed. Okay, so usually you're probably familiar with using an evolutionary tree in the context of species, but we can actually have an evolutionary tree just of different samples of one species. So here is an evolutionary tree that I've made hypothetically of HIV samples. Same exact concepts apply, it's just that your branch length units might be slightly different. Okay, so now what is an epidemic? So an epidemic, just informally we know, is the spread of some disease across a population. But for viral epidemics, we want to be able to think of this from kind of, a computational framework. So we can think of a contact network as being this graph, this network, where each individual is one node in this graph. And then people are connected. There's an edge connecting people if they have some risky contact across which the virus can transmit. So for HIV, these risky contacts might be sexual contact or shared injection drug use, stuff like that. For COVID-19, these contacts are just like, were you in the same room without wearing masks or stuff like that? It's so depending on what virus you're looking at, and specifically what is the mode of transmission, what defines a risky contact changes from virus to virus. So there's a question in the chat. In this case, the answer, oh, so this is probably from the earlier slide. So in this case, the ancestors of humans wouldn't necessarily be extinct. So so there's a slight misconception in your question. So you said like gorillas and chimps. Gorillas and chimps are not ancestors of humans. On the contrary, there is some common ancestor that like existed way back in time that is the same ancestor of modern gorillas, modern chimpanzees, and modern humans. So orangutan, gorilla, chimp, human, these are all modern day species. And there were ape-like creatures that lived way back when that were the common ancestor of all of these different species. So in both contexts, internal nodes represent things that no longer exist. But great question. Yeah, thanks for that. Okay, so yeah, so contact network nodes are people, edges, so connections between people are some interaction that those people have across which a virus can transmit. Then we have what's called the transmission network. So the contact network is just all interactions that people have across which a virus could theoretically transmit, versus the transmission network is now a subnetwork where it's the actual transmission events that did happen. So here, this person could have infected this person could have infected this person, vice versa, all of these different interactions. But in the transmission network, this person did transmit to this person at some time. This person did transmit to this person at some time, and so on and so forth. So basically, we actually have directionality along our connections, where the direction is the direction of transmission, and we have a label on each of these events, which is what time that transmission event occurred. So let's actually zoom in on one of these transmission events and hopefully tie this back to my evolution discussion that I just had. So if we zoomed in super close in a given person, we can imagine that the person that's infected, shown here on the left, has an evolutionary history of a bunch of viral sequences directly within their body, right? Each molecule of the virus evolved from some ancestral molecule of the virus, which started with some initial common ancestor that originally infected this person. At the time of transmission, we can imagine that one viral lineage, so out of all of the different viral molecules in this person, one of them transmitted and was the first infection in this recipient person. Right? So one of these viral lineages came and infected this person. This is called a transmission bottleneck. A bottleneck event, which means that basically you can think of it like a bottle, like the neck of a bottle is kind of narrow, so liquid can't really shoot through it. Only one lineage was able to make it through the bottleneck and infect the recipient. And then over time, the virus continues to evolve within the host, like the original person. And this new virus, this one lineage that went to infect this new person, has its own viral evolutionary history going on in this other person. Okay, so now imagine that these people come into a clinic and I do a nasal swab that collects some number of viral molecules from each of these person. And let's say, right, because each of these lineages represents one viral lineage that currently exists in this person. Each of these lineages represents one viral molecule that exists in this person, right? The tips are the present day species. So if I do a nasal swab and collect maybe these two molecules by chance and these two molecules by chance from this person, even though there was some complicated full evolutionary history that occurred, what I observe is basically the pruning of the full tree, because I never observed these purple branches. I only observed these red branches and theoretically the evolutionary history of those. So this is the evolutionary history of the lineages that I was actually able to observe. Okay, so there's a question in the chat. So for example, a new variant of COVID develops within one person and that person transmits the variation to another person. Yeah, exactly. So as the virus is inside your body, it's continuously mutating. I actually have a slide on that coming up. But basically, as it evolves over time, each of these different lineages within your own body are frequently mutating. And then once one lineage transmits to this one, any mutations that occurred while this virus was evolving in your body are inherited because it is exactly that molecule that transmitted to this person. So this person inherits any of those mutations that occurred down this branch of the evolutionary history. Great question. Cool, so tying to that question, so how does that actually look like? Well, we can imagine at the time of infection, at that bottleneck, there was one viral sequence, one molecule that invaded your body and became successful. And that viral molecule had some genomic sequence. There was some genome sequence of this originally infecting virus. And then over time, as we go forward in time, mutations occur in that virus. So for example, the original sequence, which we call the ancestral sequence, was originally ACG. And then over here, it was still ACG, but it kind of split down two branches. It split down this evolutionary branch and this evolutionary branch. Down this branch, the first letter, the A, mutated into a C. Down this branch right here, the C mutated to a T, and then everything below that mutation inherits it. So what goes on here is just ATG, but then maybe down this branch, this A mutated, like the G mutated to an A. So now everything below this branch inherits this mutation as well, which is why you see ATA here, versus maybe down this branch, maybe this first letter mutated to a G, which is why you see GTA here. Does that kind of make sense? So it's kind of complicated, but basically you can imagine mutations just randomly occur along each of these branches. The longer a branch is, so the more time has passed, the more mutations we expect to have occurred down that branch. Okay. Any questions about that? I know this is a little complicated. How are we able to track the mutations from bottom up? That's an excellent question. So hold on to that question because that's actually very important to what I do. But before I get to that question, let's just re-summarize this, what we call generative process, this forward in motion process. We can think of it as there's a contact network. And then if you gave me some contact network, there's a transmission network that can happen dependent upon that contact network. So along that contact network or constrained by that contact network, a series of transmissions can occur. So here we see it's constrained by the contact network because there's no connection between the green and the yellow person, which means it's impossible for my transmission network to have green infect yellow because they never interact with each other. So you can think of this as being some process that this is conditioned upon the contact network. And then similarly, the evolutionary histories are constrained by or dependent upon the transmission network. So whatever transmission network ends up actually happening strictly dictates restrictions on how the evolutionary history of the tree could have happened. And then similarly, the viral sequences evolve in a way that's very constrained by what was the ancestral sequence and what is the tree shape. So you can kind of, in a mathematical modeling framework, think of this as the contact network it can be modeled using some network model. The transmission network, we can have some mathematical model dependent upon this. The viral phylogeny can be some mathematical model where the phylogeny is conditioned upon this and sequences are conditioned upon the phylogeny. So you can think of it as like every single step is dependent upon the previous step. Okay, so now to get to Faith's question. So if I had all of this information available, so the question, whoops, yeah. So if I had all this information available to me, that would be great. But unfortunately, I can't see any of this, right? None of this is visible to me. All I can actually reasonably confidently observe are the viral sequences, right? Someone physically comes to my clinic, I obtain a sample, I run it through a sequencing machine, and I get some sequence corresponding to that person's viral sample in their body. And I'm going to talk about how people do that in the future lectures. But that's the only thing I can observe. I can't observe any of this other stuff. So that's precisely the question that we're trying to answer. The question is, given just the viral sequences, how can I work backwards? How can I infer an evolutionary history if you only gave me the sequences of the leaves? How can I infer a transmission network, or at least properties of the transmission network, if you only gave me the sequences, and maybe if you also gave me an inferred evolutionary history? And then how can I infer properties of the contact network? So maybe things like what demographics are most at risk, or who should I prioritize treatment to, stuff like that. How can I infer properties of this based on these other steps? Does that kind of make sense? So because I can only directly observe this, my goal is, how do I infer backwards from the sequences? Cool. Okay, so any questions about that? I kind of threw a lot of information at you, but hopefully it makes a little bit of sense. No? Cool, okay. So lastly, I'm going to talk about specifically what I work on, which is viral molecular epidemiology. So molecular epidemiology is the integration of molecular biology into epidemiology. So epidemiology as its own principle, you can do it based on survey data, you can do it based on like looking at medical records. There's all these different types of data that can inform epidemiology. But molecular epidemiology is specifically epidemiology informed by molecular biology. So DNA sequences, RNA sequences, protein structure, protein sequences, molecular interactions, things that are like integral to the cell, and kind of on that molecular level. This is what we're looking at and trying to infer epidemiology information from that molecular biological data. So in modern times, molecular epidemiology is largely focused on epidemiology from sequence data. So the Human Genome Project was way back when. Human genome has been sequenced, and sequencing machines have become much cheaper and much more scalable over recent times. So as sequencing costs get cheaper and cheaper and cheaper, we're more likely to keep sequencing in larger and larger quantities. And for context, the previous epidemic in which sequencing of the viral genomes was something that was interesting was Ebola, which I think over the entire epidemic, they had like hundreds or thousands maybe of viral genomes that were sequenced. And this was like mind-blowing to them. It was like, whoa, we actually got like hundreds or thousands of sequences. Versus currently in the COVID-19 pandemic, last time I checked, which was like a month ago, we were already over 700,000 sequences that were obtained from people. So we're already approaching 1 million sequences, and that's just going to be even bigger in the next epidemic. So it's really kind of like the way of the future. So just real quick, some key terms that I want to introduce. One term is epidemic, or equivalently outbreak. So an epidemic or an outbreak is a disease occurrence among a population that is in excess of what is expected in a given time and place. So for example, with the regular seasonal flu, we call it the seasonal flu because it happens regularly. So like at a specific window of time, we expect that we're going to have some number of flu cases. An epidemic or an outbreak is specifically beyond what we were already expecting. Way more of that disease is happening in a given population. So it's specifically anything beyond the kind of normative level that we would expect. Okay, so a cluster, and this is, I think people here are freshmen, so maybe you haven't taken a computer science course. The epidemiology definition of a cluster is not necessarily equivalent to the mathematical or computer science definition of a cluster, but a cluster in epidemiology just means a group of cases in some specific time and place that might be more than expected, where a case is someone that has the disease. So for example, if we're looking at the flu, one cluster would be one specific small subpopulation at the same time that this tiny group had a lot more incidence of this disease than we would have expected. So it's kind of like an epidemic is made up of clusters. Okay, so a pandemic is a disease or a condition that spreads across regions. So while it's within a single region, it's called an epidemic. And then once it spans across regions, so for example, when COVID went from Asia to Europe, Europe to US, whatever, when it jumps between populations or jumps between regions, then it's called a pandemic. Okay, and then the last term that I want to introduce is rate. So rate is the number of cases that occurred during a specific period. So for example, we have, some number of new COVID cases per day, or some number of whatever per hour, or per year. It's some number of cases during some specific period. Okay, so there's a question in the chat. Would regions be restricted to continents, or how would they be defined? That's a great question that I'm actually not 100% sure. So my understanding, at least as it was for the COVID-19 pandemic, is I think it's technically classified as a pandemic the moment it jumps from one country to another. But I'm not 100% sure if that's like the true definition. But I can get back to you if you're curious. But there's probably some like specific definition that they have. Yeah, great question. Cool. Okay, so now we've talked about molecular epidemiology. What is viral molecular epidemiology specifically? So we can actually use properties of the evolution of viruses to study a viral epidemic. So some important questions that we want to ask are things like, how did the virus spread across communities or across the world? And one tool that's been used to help answer this question is a tool called NextStrain, where you can actually kind of like plot incidences of a virus across the world. So this is from COVID-19. And yeah, you can actually like see across the world how many different, people have a given disease in a population. Whoops, I didn't want to exit full screen. That's my bad. Can you still see my slide? I hope. Let me just like type yes if you can. Yeah, cool. Okay, so then another question that we have is, what transmission clusters exist within our population? So first, the question of how does it spread across communities or across the world? That's important for knowing things like, do we restrict travel, or do we restrict dining in, or, how is the virus spreading? How do we actually intervene to prevent the spread of the virus, versus what transmission clusters exist within our population? This is more for prevention and treatment. So if I can identify what specific clusters are most at risk within my population, I can maybe allocate more resources for treatment. I can maybe allocate more resources for prevention, stuff like that. And we can actually do what's called transmission clustering, where we can infer these clusters by linking people based on how similar their viral genomes are. And then if we overlay demographic information, so here this is from actual COVID-19, if we overlay information like, do they know each other socially, or are they family members, or work, or local travel? Was it at a wedding, a temple, a bar? So this is from a real paper. Once we can overlay this type of demographic information on top of this network structure that we can infer just from the sequence similarity, then we can get some powerful information. So here it shows, whoa, it seems like most of the kind of, explosive events are happening because of social interactions. Right? Pretty much every node that has a bunch of outgoing things, it's yellow nodes that are going outside of it. So social interaction that happens like around some given source. And this can help provide information of like, oh, we should actually, it seems like not very much of it was happening from local travel. I think only this one right here. So this informs things like, what interventions make the most sense? Let me monitor the chat. Okay, cool. How is the virus mutating across the epidemic? So you've probably heard things about like the UK variant and the South Africa variant and like B117, all these terms. So basically, the virus, its genome is just some sequence, and mutations occur across that genome. And the question that we as molecular epidemiologists have is, what mutations are happening and how prevalent are those mutations? So we can actually see what is the prevalence. So these are different lineages of COVID-19 over time, I think in the US. I don't remember for sure. And we can actually see like towards these beginning stages, this yellow lineage was the most prominent. But then right around here, this dark purple lineage started becoming the most prominent. And then around here, this red lineage became the most prominent. And then over here, now this pink lineage is the most prominent. So knowing what lineages are, spreading the most in a given population is important, because then it can answer questions like, what are some of the mechanisms by which this is happening? Which is the next big question. So what are the molecular mechanisms by which the virus is invading our cells? And we want to be able to actually study and come up with these models of showing, like, so this is for COVID-19. So basically, when the virus binds to the receptors of your cell using the spike protein, it then can like bind the receptor. It has this intermediate step, and then it fuses with your cell and kind of like injects the viral genome into your cell. So knowing this mechanism can help us come up with treatments. It can help come up with vaccines, right? So this is why the vaccine for COVID-19 targets the spike protein, which is this protein on the surface of the spikes of the virus that helps it invade. Because you can design vaccines that specifically target super important functional parts of the virus. And then lastly, an important was, I don't know if, maybe this isn't the last question, but either way, another question is, are any of the mutations impacting the infectiousness of the virus? So this is the protein structure of, I think, the spike protein, if I recall. And we can actually map on mutations onto this three-dimensional structure of the protein. And then we can see, based on what we know of the mechanism and based on where we put that mutation, how likely do we think this mutation is impacting maybe how easily the virus invades our cells, or maybe impacting how easily it can avoid our immune system, or stuff like that. So it helps just give context. And then my interest specifically, which is kind of where I'm going to end off today, is, so I'm from a computer science background, and my question is, how can I translate these very relevant, actionable public health questions into formal computational problems? So how can I actually write a program that can answer these questions from real data? Cool, so that's all I have. I'm gonna maybe stop sharing there. I don't know if anyone has any questions, feel free to post in the chat. So there's a question, this question applies to the phylogeny trees from earlier, but when a vaccine is made, does it try to target the original or closest to original root of the tree so it can be effective to as many variants as possible? Not quite. That's an excellent question. But actually, kind of on the flip side, what you want to do is because the ancestors of an evolutionary tree are extinct. So even if you design a vaccine that would have perfectly targeted the ancestral COVID-19, it won't necessarily work on the present day ones. So what you want to do is actually take a bunch of present day sequences and then see what parts of their genomes are relatively stable. So what parts of their genomes are not mutating very much, and then design a vaccine that targets specifically those parts of the genome. So there's, yeah, no problem. Yeah, great question. So there's a question in the chat. When will the week 1 reflection be posted? So it's actually already on Canvas. So if you go to Canvas, you should be able to submit the week 1 reflection by just clicking here and then clicking submit assignment, and then just like typing your reflection. You can, I think you just can type. Yeah. So that's already up. Cool. Okay, so there's a question in the chat. How do you tell what parts of a virus are stable? That's an excellent question. Let me actually pull up a figure that I have. Let's see, who did I send it to? I sent it to someone recently. Who was it to? Sorry, there's like one specific figure that I have that really shows it. There we go. And I'm gonna maybe post a link in the chat. There we go. So basically, so this is a public website run by Los Alamos National Laboratory. And this specific page of it is about the spike protein of COVID-19. And the horizontal axis here is position in the genome. or like position of the protein. So this is the beginning of the protein. This is the 200th letter of the protein. This is the end of the protein. And on the vertical axis is what's called entropy. I'm not going to go into a bunch of details of what entropy means, but basically high entropy means high variability and low entropy means low variability. So what this means is parts of the protein that have low entropy are parts that aren't really changing across different people in the population. So what you would do is, like, let's say like this segment of the protein is super, super stable. I could design a vaccine that specifically targets this chunk of the spike protein. Does that kind of make sense? So how do we know if a variant has gone extinct? There's kind of two parts to the answer to that question. One part is how do we, so basically that kind of the simple answer is if we keep sequencing people and we keep looking for the variant, but none of the people that we sequence has that variant, then we would potentially say the variant maybe has gone extinct. The problem with that is that we don't sequence everybody that has COVID-19. There's sampling bias, there's subsampling of the full population. So the problem is, even if nobody that we're sequencing has that variant anymore, the question that comes up is, does that variant not exist anymore? Has it gone extinct? Or does it still exist, but we just accidentally didn't find people that have it? And the answer to that question is a little bit harder. So the TLDR is I don't really know. And I don't think we can ever really be 100% certain. Cool, yeah, any other questions? We got like 1 more minute. If people want to like unmute themselves, feel free to raise your hand and I can set it to unmute. If you want to like ask it verbally or. Nice, there's another question in the chat. Since there are still some mutations on the stable parts, will vaccines account for minor variations? That's a great question that I'm unfortunately not the most knowledgeable about. So I don't know too much about vaccine design beyond just, you know, what parts of the genome might be good candidates. So one thing that I do know is that they try to design vaccines to have a little bit of flexibility. So even if just one part of it is different, the antibodies your body generates should still be able to detect it. And in the clinical trials that they do to determine the efficacy of those vaccines, they try to get as wide of a sampling as possible. So the hope is, even if we don't necessarily know the molecular mechanism of is it still working or not, we would at least kind of at a global scale see we had all these people from all these different lineages and the vaccine was effective in all of them or something like that. So there's a question in the chat. I'm a little confused. What are the expectations for the reflections? So basically, so the reflections are just my alternative to taking attendance. It's just really like write a paragraph or so of something about that week's topic that you learned or found particularly interesting. There's not really any expectations beyond just completing it. Cool. Nice. So I don't want to take too long because we're at 1:50, but yeah, thanks everyone for showing up. Basically, each week I'm going to be covering one specific step of a standard viral molecular epidemiology workflow. So yeah, I'll see you all next week. If you have any questions, feel free to post on the discussion board. Feel free to email me. I'm happy to help. Bye, everybody.